weird perhaps is, I don't know, if you really had a lot of pain and you had a lot of different medications for it, maybe you also, and that's often for localized pain by the way, you sometimes would maybe have heard about capsaicin as a pain treatment, which is a little bit counterintuitive, because if you would apply capsaicin to the skin, it would be painful because you activate these receptors. It's not nice to have a pain, and still people with chronic pain and they give these capsaicin patches or something. It's a little bit weird perhaps, but the main reason is that if you activate these C fibers, as we call them, so these primary afferent fibers taking the signal to the spinal cord, that if you all the time activate them, because there is capsaicin around, so all the time to the spinal cord, and in the spinal cord we get synaptic communication between this neuron that is active all the time and the spinal cord neuron. And in that communication you use neurotransmitters which need to be produced all the time. So if you keep on having the capsaicin signal, at a certain moment we ran out of neurotransmitters, so we depleted the neuron. So that means that we are for still quite some time before actually the neurotransmitters would be replenished. We are without signaling. So although there is an initial pain, after that you have a period where there is supposedly no pain at all.
But at the end, new channels are being made.
Yeah.
So does it work on long term or short term?
No, it's temporary. Probably not attach after.
Okay.
Is it possible to apply the gate control theory on demo receptors and acid receptors as well, or only to?
On which receptors? Sorry.
The gate control theory.
Mm-hmm.
Can you apply those to those acid receptors and chemoreceptors as well, or only to mechanical?
In theory, yes, but we will come back to that. Maybe you keep that question if it still is there after. So this is something just about ionotropic receptors. So you see that this is actually our ion channel, and it can be activated either by a ligand, as with a TRPV1, or it can be activated by a voltage difference, as with these sodium channels that you saw, which are voltage operated. So, if you think about an ion channel like this, what goes through an ion channel? Yeah, which ions? Because you have different ions. You have
Sodium, calcium.
I hear all kinds of different things. That means that every ion channel will be permeable to all these different ions.
No.
What does it depend on?
The structure of it.
Yeah. So it depends, for example, on the opening, the size of the opening. It can also depend on, let's say, the charge in the pore, whether there is a lot of negative charge or positive charge or whatever, what would fit through. So you have all different kinds of ion channels. You have ion channels which, for example, mediate the flux of anions, like chloride or something, which are negatively charged, also positively charged. And size matters, so not everything fits through. Okay, then the spinal cord dorsal horn. So here we see the same fibers as we talked about before, coming from the periphery and entering the dorsal horn. So this is CNS. And you see that there is very specific innervation patterns. Not every fiber ends up in the same part of the spinal cord. You have some fibers which are ending, let's say, more in the superficial dorsal horn, because we call this whole thing, let's say, one of the upper wings of the butterfly, we call that the dorsal horn. And then, of course, we have a second dorsal horn on the other side. The ventral horn would be the lower wing. And ventral horns contain those motor neurons that we talked about. But in the dorsal horns, we have the sensory innervations. A question for you. Where would these circles or whatever, where would they be located? In the? Yeah, in the dorsal root ganglion. So remember that we had that bulge in the dorsal root. It is only extended in size simply because it contains all those cell bodies, and they of course need a little bit more space than just the fibers. That's actually why you have that ganglion in the dorsal root ganglion. And the fact that you don't have a ventral root ganglion already would tell you that there is no cell bodies in the ventral root. So they are located, like we said, in the ventral horn of the spinal cord. Anyways, we have different fibers from the periphery. We have so-called C fibers. You see two specific types, non-peptidergic, peptidergic, but forget about that. C fibers and A delta fibers, but we also have A beta fibers. And the main difference is that the A beta fibers, which you can see, they come in here, they travel, and they end up a little bit deeper in the dorsal horn. These are the fibers which mediate touch. And there is something that is not fitting in my story. I said something that goes against my previous story. So what I say here, I have touch fibers which come in, and they innervate cells in the deeper dorsal horn.
And you said before that it's directly going up to
Exactly,
the brain cell.
exactly. So I said before, a touch fiber normally enters the spinal cord, then it ascends immediately the dorsal columns, and the first synapse is in the brainstem, which is, by the way, totally true. Except that when we are talking about, so actually what this cell would do, it would go in here, it would project into the dorsal column, project towards the brainstem. That's what it also does. It's not indicated in this picture because when we are studying, we like to simplify the things. What we're looking at here is a collateral branch. So it's not the main nerve axon. It is only a collateral branch, a side branch. The reason why touch fibers have a side branch, so only here you can see visualize the side branch. So the main branch would actually travel into the dorsal column up towards the brainstem. The reason why we have a side branch is because touch can modulate the nociceptive signal. That's also what you probably know, that when you hurt yourself, you start doing that and you feel less pain. It's because we have this side branch, and that we call the gate control theory. We call it, who did not, just be honest, because there is no shame or whatever, who did not hear about gate control theory? Because then I will explain a little bit more. One hand is not good. So, the gate control theory is one of the oldest pain theories if you talk about modern science. There were two scientists, Pat Hall and Ron Welte, who came up with that, and they said, if we have something like that, and how to explain that? Let's come up with a model. The model should contain a neuron. Should contain maybe another neuron, because we have touch and we have pain, two neurons. And they start coming up with a scheme, like how could they be connected to explain the phenomenon. We will see that later, by the way, because it's important to slide. The main reason is that they stated, you will see that later how that would work in the scheme, they state that the spinal cord, dorsal horn, somehow contains a gate. Just like you would have a castle with a big gate in front, and the gate is closed, and nobody can enter the castle. If the gate would be opened, you go into the gate, through the gate into the castle, and the castle ends up in the brain and says, ah, it hurts. So either the gate is closed and we don't feel pain at all, or the gate is open. So there should somewhere be a gate in here. One neuron, if active, into the castle and it hurts. If not active, then the gate remains closed, we can't enter. So there is no pain sensation towards it, no second towards it. Now, you will see later on that actually these A beta fibers, because they are of course the fibers which are running this signal, that they will connect to interneurons, such as this one, PKC gamma interneuron. And these neurons, it's not indicated in this figure, but will be when we are talking about the gate control, they are giving an inhibitory signal to the gate. So like you have gatekeepers on both sides, and when they see the inhibition, they say, no, we keep the gate closed. That's why when you do this, you actually keep the gate closed and there is no pain signal. That's why it hurts less. But you will see that later, a little bit more detail, and there is no castle in those pictures.
Was it also, I thought they modulated the signal on the fibers themselves.
Yeah, exactly. That's how it
How
works.
about to inhibit?
Very good, but we will see it later, but you're right. Okay, so if you look at it, you will see A beta fibers, they are going deeper, but the A delta and C fibers, which are actually the nociceptors, so they are the ones connecting the signal about, let's say, harmful stimuli, they are ending up in the superficial dorsal horn. You see, you have all a little bit superficial, lamina 1, a little bit here. So that is where normally a signal would arise. If this spinal cord neuron would be active, there is a signal towards the brain and we say, oh, that hurts. That all happens in the superficial dorsal horn. So the deeper dorsal horn, in a way, would only be there to modulate the activity in the superficial dorsal horn, so that we would feel less signals going to the brain and saying, ouch. That's the basic concept. Now, did you already do immunohistochemistry in the...
Yeah.
So if you talk about these kind of topics, you can study them immunohistochemically. You can say, so we really have that organization. Okay, let's visualize that, because we are scientists after all. Exactly. I have the same idea. So how would you do that? Suppose that you say, okay, we have an ABET fiber, we have a C fiber. You see, we have different types of C fibers. We have peptidergic, which end up totally laminar 1, and non-peptidergic a little bit lower. How would we take advantage of what would we take advantage to visualize them? Well, let's say, what do you usually do when you do an immunohistochemical staining? What do you stain for? Specific what?
Protein.
Yeah, usually specific proteins indeed. So you just say, oh well, if this body expresses protein A, I simply run a protein A staining, and I will see only those guys. And if these ones express a different protein, I'll say stain for that protein. That's what we did. First, the C fibers. Peptidergic C fibers. What would we stain for? Just give it a guess. A peptide, exactly. Yeah, we take a peptide. So the peptides expressed here are, for example, something we call CGRP, calcitonin gene-related peptides. It's a peptide, which is expressed by these peptidergic C fibers, and the peptidergic C fibers are really in the superficial, superficial dorsal horn. And here you see it. So you have the spinal cord of a rat. Okay, ipsilateral and contralateral, forget that, because this is after a nerve injury in the periphery, but that we will not discuss here. So you see stainings which are really in the superficial dorsal horn. And you also see some staining going down. That's because C fibers do not only project to the superficial, they also have some branches deeper into it, but not so many. But you can see them. So that's all using immunohistochemical staining for CGRP. You see peptidergic C fibers. Then non-peptidergic C fibers. Well, that's a little bit tricky, so that I will help you with. You also have in membranes, you can have certain carbohydrate chains. You know, you have proteins, you have carbohydrates, you have, of course, the lipid bilayer. But these fibers specifically express a glycoprotein that you can visualize using an isolactin. It's like a sugar chain. And this is isolactin B4. Forget the difference between left and right, because it's again in the pathological model. But here, with a little imagination, you would see, compared to the other one, that this one runs a little bit deeper, and it's a little bit less intense in the upper layer of the dorsal horn. That's because you have that just a little bit lower innervation pattern of these fibers. So that's the non-peptidergic C fibers. And then finally we have the A-beta fibers, and those ones can be specifically stained by using an antibody which is directed against a protein, of course, that is only expressed in A-beta fibers, and this protein is a protein involved in transporting neurotransmitters into vesicles so that they can be released. But you have all different kinds of transporter molecules for that. So what I mean is, if you have a terminal of an A-beta fiber in the spinal cord, and you have all kinds of neurotransmitter vesicles in there. But to get the neurotransmitter into the vesicle before they can be released, you need transporter molecules. And you have all different kinds of transporter molecules. And the transporter molecule that is specific for the A-beta fiber is V-GLUT1. And if you do a staining like that, you see this. And you see with a little imagination that you have a very vague area here, because you do not have a lot of innervation of those E-beta fibers on the superficial, but it's more intense to the deeper dorsal horn. Now you also see here, probably, that the staining qualities differ from one staining to the other, although the secondary antibody that we use in all these stainings is identical. So this is something about, that is also one message if you do an immunohistochemical stain, that each primary antibody needs to be tested first. This is actually one of the better ones that we have, but you could say, theoretically, either there is a lot of A-beta fibers everywhere, or there is a lot of background, let's say a non-specific staining, because the antibody maybe is less specific for the V-GLUT1, but also recognizes a little bit other proteins in the tissue. And we have a signal that we actually are not interested in. But this is something to find out, which is not really the case for this one. But you see different kinds of qualities of stainings in these kind of researches. So that also probably means that if you want to give a number to the expression, whether or not something increased or decreased, that you're often dependent on the kind of quality images that you get. So techniques are never 100% waterproof. But yeah, that is. And the last issue, and then we do the break. What is our time? Still. So we still have 15 minutes. Huh?
Yeah.
Good. Of course, there is not. Your brain is not only equipped with systems to get a signal towards the brain and saying "ow," but also at certain moments been enough. So you have, let's say, inhibitors of the nociceptive signal, which come from the brain itself. One is the gate control theory, as we said before. So I tried to visualize it in these pictures. Let's say if you want to understand the nociceptive system, probably you will first say, sensation and touch and pain, it's kind of the same thing, maybe a little bit different interpretation, but it's a signal, sensation that is in the periphery and I feel it. So, in the past, people also, and in the past, and I'm talking, for example, the beginning of the 20th century, so 1900, I don't know what, somewhere in the beginning, they were saying, probably you will have one system which is for touch and another system which is for nociception. So one ends up saying touch and the other out towards the brain. Really, as they say, parallel lines, labeled lines. You have a labeled line for touch and a labeled line for nociception. So the blue one would be the peripheral neuron, and the white one would be in the spinal cords, projecting towards the brain. Right? Ow, oh sorry, here, ow, and there, no. I feel it. Okay, this concept. And then he said, yeah, but maybe it works different. Maybe it goes like that. We have theory of pain on convergence. So we have at least two separate fibers, because of course you need to have a discrimination between what is damaging and what not. But then in the end, in the spinal cord, they end up targeting the same neuron and then goes to the brain, and you have a mix between touch and nociception. But this is a little bit counterintuitive, because I'm sure that you can discriminate between something that you feel or something that hurts. But still, so let's say normally the intuitive concept would be that you say, yeah, probably we have this. But the support for the convergence theory came with this gate control theory. And it was simply like when you do this, you feel less pain. So it's impossible that you have only this. At some stage, you should have a convergence, because otherwise this could never influence the nociceptive signal or pain. So that was the gate control theory they came up with. And like I said, it is still called a gate control theory, because a theory is true until you prove false. But there is no unequivocal data to show that actually there is something like this. It's all just a concept. It's the best concepts we have to understand why this works less. That's it. Whether or not all these things are true, we don't know for sure. The gate control theory states that you have this kind of convergence. We call this neuron in the spinal cord a WDR, wide dynamic range, simply because if you look at the figure, you will see that it gets input from a nociceptive fiber and from a touch fiber. So that means that this neuron should be responsive to touch and to nociception. So that means, suppose that I have a pipette into the spinal cord, touching this cell, containing an electrode so that I can measure the electrical currents in that neuron. And I would now give a stimulus to the skin, the area of the skin that is within the receptive field of this neuron, and I would give brush. Then that should lead to activity in there, because it receives input from touch fiber. If I would give a pinch, really painful pinch, you also see some activity in there because it receives information from a nociceptive fiber. And those neurons have been demonstrated in the spinal cord. So there is neurons which respond to both, as we call it, innocuous stimuli, so touch stimuli, not hurting, and also to nociceptive signals. So we have those wide dynamic range neurons. So it would be nice, simply the fact that we have that, because probably they are important in gate control, because we also need to have neurons which receive input from nociception, but also because this hurts less. And then the whole idea is, so that's to make a long story a little bit shorter, that we have a pain signal Nice, I made it up. But you can also have at the same time, because there is an interneuron in the spinal cord, that's what you saw before, that PKC thing there in the spinal cord, that is in between, and this interneuron is, and then, Aaron, I think you were the one making the remark that you actually got an inhibition before it reaches the, so that's true. You have an interneuron that makes a synapse with these primary afferent fibers at presynaptic locations. You see? When you look at the nociceptor, the nociceptor itself makes a synapse right here. The non-nociceptive fiber, touch fiber, makes a synapse here. But the interneuron kind of, let's say, makes a synapse at a presynaptic, just before the synapse, presynaptic site. So it can intercept, in a way, the signal, if it would be active, because it's an inhibitory signal. And if you remember well, any signal going over an axon is just an action potential that's mediated by voltage-operated ion channels. So suppose that I get an inhibitory signal in that axon membrane, actually the action potential will diminish, gone, before it hits the terminal, so there is no neurotransmitter release. So what the theory then states is that if I got an active, like rubbing the hand, intercepts, and doesn't reach the signal doesn't reach anymore the spinal cord, and there is no, the gate stays closed. Like that. So let's see again.
And I'm proud of your animation.
So you should be proud of something. Anyways, so that's the main concept here. That is one way to inhibit the pain. So this is, let's say, if you talk about systems, what is responsible for inhibiting the nociceptive signal here? Which mechanism would you say, again, gate control, but let's say, which cell is responsible for the inhibitory signal? The interneuron. A very localized neuron. Of course, it's activated by touch, but the one really inhibiting is that yellow cell here. So this is a very local inhibitory circuit. It's really there in the spinal cord where the whole signal is. It can close the gate at that level. But of course, you know that you can probably feel less pain, not only by doing that. You have another situation that you say, would that happen? Probably also feel less pain. I can mention one, but my example is not so nice for the girls because they don't feel the connection. And I like to play soccer. And I was once in the Nou Camp Stadium, Barcelona, so a huge stadium, and I always imagine, like, suppose that you would be one of these gifted players like Messi, entering the stage, and maybe, you know, scoring one of the most brilliant goals, and the whole stadium, and your name and stuff. I mean, what kind of an excitement must that be? And suppose, because that's what you often hear, that in case somebody would maybe, I don't know, twist an ankle or something, really have a severe injury, that they don't even notice that they actually play still for a while, and then when they maybe calm down a bit, they actually start feeling the pain, and oh, something is wrong. So, I mean, for girls, maybe it's when you have, I don't know, maybe a symphony in a huge theater, or...
It's a little bit sexist. There are also girls that like soccer. Even the boys that don't like it.
Sorry about that. You're totally right there. I take it back completely. Anyways, you get the idea. So that means this is something. This is an inhibitory signal, of course, that cannot be explained by a very local interneuron, because probably it has something to do with your brain activity. You're excited. Anyways, so that means we are looking for something else. And of course, you heard about these kind of peptides that you can also administer yourself from external sources, but actually the body itself. That's actually also a nice story. That when you talk, I mean, everybody, for example, probably knows, I don't know, morphine or something like that, or heroin or stuff like that. But the thing is that all these drugs, when you think about it, they discover the drug, and when they're doing science, actually they start seeing that, oh, we also have receptors for these drugs. And that's actually quite weird, because why would a body have receptor proteins for a drug that you find in nature? So probably there is something in the body itself like that. And then they found the endogenous opioids. So we all have our own morphine. Our body produces own morphine, which we then call endorphine. It's very original. Not really. And the whole concept about that is that if you have a lot of excitation, then actually you are activating centers in your brainstem, which can send projections down to the spinal cord, back to the gate again. And they are inhibitory. It's what we call descending inhibition, because it descends from the brain to the spinal cord, and it kind of also closes the gate. Just like you would have an interneuron in the spinal cord, you can also have a descending system from the brain, which inhibits the gate. But it's activated by another. It's not because we are robbing some part of the brain, but simply because we have these opioids active. Opioids are active because we are excited or whatever. And how do they modulate the system? That is something not really to remember, I think, but I just put it in so it makes more sense of the story. If you have these mu opioid receptors, they can either inhibit the calcium channels, and calcium is, of course, an essential molecule to have neurotransmitter release, because you need to have first calcium in someone, the vesicle releases the neurotransmitter into the cleft. So that occurs far less now because it's inhibited, so you have less communication. And also, on the postsynaptic cell, you actually increase the conductance of potassium channels. And potassium channels keep the cell hyperpolarized. So there is less chance of a depolarization, also less chance for signal production. So it's just the two processes that are mostly allocated to opioid function. Okay. Opioids usually work because, let's say like that, what do you know about an opioid? How does it travel, an opioid? Yeah, it's a hormone, so it travels in the blood. It can have anywhere, it goes through lipids, goes through everything. Now, when I say it activates descending systems, that doesn't really match to this story, because here I see opioid receptors already in the spinal cord. So why would I need to have a descending inhibitory system when I already have an opioid? I can simply have opioid production in the brain. The opioid ends up also everywhere, also in the spinal cord, and inhibits the signal here. I don't need to have descending pathways, which is true. However, if you look at the same areas in the brainstem which are producing the opioids, you see that they also have projections down to the spinal cord. At the moment that you get opioid release here, the opioids indeed go into the blood, and they also end up in these areas, inhibiting the whole signal production there via their receptors. But at the same time, these opioids also activate the neurons here, which are sending inhibitory signals towards the spinal cord. So actually the action of the opioid is twofold, via descending modulation, but also via simply the action of an opioid on its receptor in the spinal cord. How do you know that? That's also very simple research and, with cats, and I don't know what they did in the past. They simply make sure that there is opioid expression, then they cut the connection, the descending connection. You see that a lot of the inhibition of the pain signal disappears, showing that for the proper 100% functioning of an opioid, you also need descending firing systems, which are given this inhibitory state. An important neurotransmitter for the people interested, which is involved in that descending inhibition, is called serotonin. You can imagine that if you want to modulate something in the spinal cord, that you see the dorsal horn, there is a lot of cells in there. We have a superficial dorsal horn, we have a lower dorsal horn, deeper dorsal horn. Now suppose that you have a system like that. It comes from the brain. 2 minutes. Oh, a minute. Suppose I have a system coming from the brain and it needs to inhibit almost completely the dorsal horn. Can I then say, if I have such a modulatory system, well, I let these fibers run in a specific tract, and the tract is located exactly here in the white matter, and then when the axons come out, they go here to that cell. Would be possible, right? I mean, theoretically, yes. But when I tell you that a modulatory system coming from the brain, which is actually the only thing it's modulating is neuronal activity. It's not at all specific for sensory processing. I also, for example, have serotonin coming from brain areas like the heart, the same area, and they are modulating motor functions, the motor cells in the ventral horn, which is serotonin, for example, is one of those essential mediators to make sure that I do this coordinated. One, then left, then right. Without serotonin, probably it will totally be disorganized. So serotonin projections from rapid are really over the entire spinal cord. That's why these modulatory systems, like the serotonin system, is not at all specifically located to attract, but it's very diffuse. You have fibers everywhere in the... So if you would do a serotonin staining, you would see something everywhere in the white matter. You don't see a bundle in the white matter. The reason why I mention it is that we call this system of inhibition, we call it diffuse noxious inhibitory control, DNIC. Diffuse noxious inhibitory control. So it's a control which is diffuse, it's inhibitory, and that is there when there is a noxious stimulus. So suppose you have a terrible pain in your ankle because you twisted it. But in time it gets better and better. It's probably because you're activating these descending systems from the brain. It's all the time inhibition. It's probably you know it as habituation to pain. But it's simply because the brain is telling you, it's good enough. That's inhibited signal, we know it now. Except that there is a real disease going on, then things can get out of hand, we get chronic pain. But in normal conditions, when you get habituation to pain or whatever, it's because of these kind of descending inhibition. DNIC, diffuse noxious inhibitory control. That's basically it. So you heard something this morning about anatomy, something about how a signal is communicated from the peripheral side to the brain. You also heard something about how the brain and also spinal cord mechanisms, how they can modulate that signal, so you get an inhibition of pain. That's basically it. And I think that you should regard this first lecture, and the questions were okay, but I can expect that you maybe have far more questions, so don't feel any inhibition. Anyway, so that because this is just the introduction actually, and I think the more, I would say, more exciting is coming after the 30-minute break, and that will be about chronic pain conditions, why people have pain where they're not supposed to have pain, and what can we do about it. So that's maybe something that you can apply probably to your own environment as